Hello everyone, and welcome to today's session where we're looking at gene expression and we're going to look at transcription. When you're talking about gene expression, you're talking about transcription and translation, and these are the means by which cells read out or express the genetic instructions in their genes. And during transcription, DNA serves as a template to make a messenger RNA molecule, and this occurs in the nucleus. Thereafter, the messenger RNA will then leave the nucleus and go to the ribosome. In the cytoplasm, where translation occurs, and translation reads the genetic code in messenger RNA and makes a protein. So let us look at transcription, and this is a transfer of genetic instructions in DNA to messenger RNA. And so we see during transcription, a strand of mRNA is made, and it has complementary base to a strand of DNA. Like in replication, we see that synthesis of an RNA occurs or takes place in 5 to 3' direction. And the strand of DNA that is transcribed into RNA is called the template, whereas the other strand is called the coding strand. In eukaryotes, we have 3 different types of RNA polymerases, which catalyzes different RNAs. We have the RNA polymerase 1, which synthesizes ribosomal RNA. Polymerase 2 will synthesize messenger RNA, whereas polymerase 3 will catalyze the formation of transfer RNA. So let us look at the steps involved in eukaryotic transcription. We start with initiation, and we see that RNA polymerase requires other proteins called the transcription factors to first bind to the promoter region and then recruit the correct or the appropriate polymerase. And so the first event, initiation, is a binding of transcription factor 2D protein complex to the Tata box via one of its subunits, and that is the Tata box binding protein. So as soon as TF2D complex has bound, TF2A binds and stabilizes the TF2D and Tata box interaction. Next, TF2B binds to TF2D, and then RNA polymerase, which already has complexed with transcription factor 2F, now binds. And this will be followed by the binding of transcription factor 2A and H. And so we'll have a final protein that will protein complex that will contain at least 40 polypeptides, and it's known as the transcription pre-initiation complex. Other proteins that will bind this complex include the activator proteins, which increases the transcription rate, the repressor proteins, which tend to decrease the rate of transcription. We also have enhancers. These are DNA sequences that boost transcription by binding to the transcription factors. And then we have the mediators, which act as a bridge between enhancers and the promoter region to regulate transcription initiation. When it comes to the elongation phase, we have TF2H carrying out 2 functions. One, it is a helicase, and so it utilizes ATP to unwind the DNA helix, so allowing transcription to begin. It also adds a phosphate, or it phosphorylates RNA polymerase, and this enzyme, it changes its conformation and it's able to dissociate from other proteins in the initiation complex. And the region that is phosphorylated when it comes to the RNA polymerase 2 is a long C-terminal called the C-terminal domain of the RNA polymerase 2 molecule. It is important to note that only RNA polymerase 2 that has a non-phosphorylated CTD can initiate transcription, but only an RNA polymerase II with a phosphorylated CTD can elongate RNA. So RNA will then start, RNA polymerase II will then start moving along the DNA template, synthesizing RNA. And this will take place in the 5' to 3' direction. The final phase is the termination, where we have the terminators. And you find that termination of transcription is different for the 3 different eukaryotic RNA polymerases. When it comes to the rRNA genes, they are transcribed by RNA polymerase I. They contain a specific sequence of base pair, which is recognized by termination protein called the transcription termination factor for RNA polymerase I. So the proteins will bind to the DNA at its recognition sequence and block further transcription. And this will cause the RNA polymerase to disengage from the template and release the newly synthesized RNA. When it comes to the RNA polymerase II, it does not terminate transcription at a specific time. However, it can stop at varying distance downstream of the gene of interest. So RNA polymerase II can continue to transcribe RNA anywhere from a few base pair to thousands of base pair past the actual end of the gene. And so this is what happens. Remember the extended tail of CTD of RNA polymerase II? It will recruit proteins for polyadenylation. And these 2 proteins are the cleavage and polyadenylation specificity factor, that is CPSF, and cleavage stimulation factor. And when the RNA polymerase II reaches the end of the gene, it will encounter an upstream of polyadenylation, which has AAUAA sequence, and a downstream, which is rich in the guanine uracil-rich sequence. And these sequences are separated by 40 to 60 nucleotides in the emerging mRNA. So once this sequence have been transcribed into the messenger RNA, CPSF and CTSF will bind to the newly synthesized messenger RNA. So we find that CPSF will bind to the AAUAA sequence, whereas the cleavage stimulation factor will bind to the guanine uracil-rich sequence of the emerging mRNA. To these, additional cleavage factors will be recruited, and it will result to the cleavage of messenger RNA, and this will occur at a site 10 to 30 nucleotide downstream from the polyadenylation site. We then have the polymerase enzyme catalyzing the addition of a 3' poly A tail on the pre-messenger RNA, and this is recruited by the CPSF into the cleaved RNA. So the enzyme will add about 200 poly A to the 3' end, which the adenine and the source of adenine is ATP. Important to note that RNA polymerase II does not dissociate immediately when you have cleaved the pre-mRNA and polyadenylated it. It continues to move along the template, generating a second RNA molecule. And so the remainder of the transcript is digested by a 5' exonuclease while it is being transcribed by the RNA polymerase II. And it is degraded, or the exonuclease degrades the mRNA from the 5' end. So when the 5' exonuclease catches up to RNA polymerase II by digesting away all the overhanging mRNA. It helps to disengage the polymerase II from the DNA template and therefore bringing transcription to halt. Messenger RNA is synthesized in large precursor form known as the heterogeneous nuclear RNA or pre-mRNA, and therefore it must undergo post-transcriptional modification. And this includes the 5' end capping, and this capping takes place in 3 steps, and each step is catalyzed by a different enzyme. And because these enzymes are bound to the RNA polymerase in the long phosphorylated tail, we see that mRNA capping occurs during transcription in the nucleus. So we see that the messenger RNA is first synthesized with a 5-triphosphate group. And so the first step in capping is to remove 1 phosphate. And this reaction is characterized by RNA triphosphatase, which removes a single phosphate, resulting to a 5' diphosphate terminus. The mRNA 5-diphosphate then binds to a GMP, that is guanosine monophosphate, and this reaction is characterized by RNA guanylyl transferase, which forms a triphosphate bond with the new guanosine. Thereafter, through the action of guanine NS methyltransferase, the molecule is methylated at the NS-guanine base. And so what is the significance of the 5' end capping? 1, it enhances translation, as we shall see. 2, it protects the messenger RNA from some ribonucleases. And 3, it enhances transport of the messenger RNA from the nucleus to the cytoplasm. And here is just an example of a 5' end capped messenger RNA. The other form of modification is a polyadenylation at the 3' end. And you see, polymerase is also responsible for the addition of poly-A tail at the 3' end of pre-messenger RNA. The other modification is splicing. The messenger RNA or the pre-messenger RNA that has been synthesized does not contain a sequence which will be needed to form protein. Some regions will not contain the gene of interest, and these are known as the introns. So they do not have or they do not contain any genetic information, and therefore they have to be spliced. The genes that contain the genetic information are known as the exons. So splicing removes introns and joins exons to form a functional messenger RNA. And so it requires special proteins and also small nuclear RNA, which consists of about 100 nucleotides. So we see that the base composition of the small nuclear RNA is complementary to the ends of introns. And so this will help the small nuclear RNAs and introns to base pair, and this leads to formation of a loop. And as a result, the exon come together. So finally, the loop is removed and the exons are joined together. We also have what we call alternative splicing of messenger RNA molecules. And we find that the pre-mRNA in human can be spliced in alternative ways in different tissues. And this will produce multiple variations of messenger RNA, and therefore its protein product. And this mechanism will lead to a large and diverse set of proteins from a limited set of genes. We have an example of the messenger RNA for tropomyosin, which is an actin filament binding protein of the cytoskeleton. It undergoes extensive tissue-specific alternative splicing with production of multiple isoforms of the tropomyosin. And so with that, we've come to the end of this session where we have looked at transcription. We have looked at the three phases, that is initiation, elongation, and also termination. Thank you.